Alright class, welcome to video 3 of section 10.2, events in the cell cycle. This is Mr. Worley here. Let's just go back and check our objectives, which are right here. Name the events of the cell cycle. Name the main events of the cell cycle. Okay, so we've got interphase, then we've got interphase, and then we've got M phase. And in interphase, we've learned about the G1, we've learned about S, and we've learned about G2. Now we're going to learn about M phase, which is the mitosis. Check it out. Describe what happens during the 4 phases of mitosis. What is phase? Phase means part, or portion of. Alright, so let's go to the next page on 246 and 245, I think. No, 246, 247. And this is what this page looks like. Basically, this is a big overview of the cell cycle. Here, what we just read about is interphase. That's when G1, S, and G2 happen. The cell's growing, it's copying its DNA, it's getting ready to divide. Now we're going into mitosis. Mitosis is this part of the circle right here. Mitosis is this part of the circle. It has 1, 2, 3, 4, 5 steps, and these 5 steps happen quickly, and that's why they're small on the pie chart. But they're important, and I want you to know exactly what happens in each one. So let's read together. I recommend that as we read, you put it on pause and just look over the book and the picture to make sure you understand each step of the process. All right, here we go. The first one is prophase. Mitosis. Biologists divide the events of mitosis into 4 phases: prophase, metaphase, anaphase, and telophase. Depending on the type of cell, the 4 phases of mitosis may last anywhere from a few minutes to several days. As you read about each phase of mitosis, look at figure 10.5. First, prophase. Okay, let's look at figure 10.5. Prophase is right here. Chromatin condenses into chromosomes, the centrioles separate, and the spindles begin to form. The nuclear envelope breaks down. Okay? So back here in interphase, the chromosomes are, the DNA is copied, but they're not in chromosomes. It's all kind of this cloudy mix called chromatin. But in prophase, the chromosomes bundle up, tighten up, and become actual chromosomes with the centromere and the chromatids. Alright, what else is happening? Spindle fibers are forming, and the centrioles are getting in action. Let's read. Prophase. The first and longest phase of mitosis, prophase, can take as much as 50 to 60% of the total time required to complete mitosis. Oh, that's a great quiz question. The first and longest phase of mitosis. Also, during prophase, the chromosomes become visible. The centrioles, or 2 tiny structures located in the cytoplasm near the nuclear envelope, separate and take up positions on opposite sides of the nucleus. Alright? And that's exactly what happened. Here are the centrioles. They take up positions on opposite sides of the nucleus. The nuclear envelope breaks down, and the chromatids condense. I want you to know what happens in prophase. You might want to write in your notes, just draw a circle, just like you see here. And if you can, draw a little nucleus and just draw this little picture here and see if you can get it. Okay, my art obviously isn't very good, but I'd like you to know what this is, prophase. Okay, let's keep going. Page 247. The centrioles lie in a region called the centrosome that helps organize the spindle, a fan-like microtubule structure that helps separate the chromosomes. During prophase, the condensed chromosomes become attached to fibers in the spindle at a point near the centromere of each chromatid. Interestingly, plant cells do not have centrioles, but still organize their mitotic spindle from similar regions. Near the end of prophase, the chromosomes coil more tightly, and in addition, the nucleolus disappears and the nuclear envelope breaks down. Okay? Now let's move into metaphase. Please go to the next video.